All right, well, thanks again, and hi, everyone. It's so nice for this invitation. We don't have a lot of time today, so my goal is really just to give you a broad overview of the technologies that are available through the genomics facility, as well as some applications that folks are doing using these instruments. I know many of you are already using our services, but perhaps you'll find there are other areas in which we can be of assistance. I'm sure, like many of you, I'm working from home today. This is my coworker, Odin. I thought it would be fun to invite him to a meeting once, but that backfired, as you can see. So you may be wondering if I'm at home, well, who's doing all the sequencing? So I just want to start by acknowledging my colleagues. I'm so grateful to be part of this team. Lily and Leanne are really the ones keeping the lab running right now. Many thanks as well to our bioinformatician, Jake, and our director, Dr. Mike Surrett. And if you ever need to contact us, feel free to use any of the email addresses at the top of the slide. We have a couple lab-specific ones, and I've got my email address up there as well. So ours is a service lab. We're really here to help all researchers and lend our expertise wherever we can. Even if you just have questions and won't necessarily be using our lab services, we'll certainly do our best to try and help you. In case you've never been to our labs, we're located inside the Farncombe Institute, which is on the 3rd floor of the McMaster Hospital. Brand new entrance. And inside the facility, we actually have 4 separate lab spaces. And this division is really important for contamination control, as so many of our workflows involve amplifying DNA by PCR. So for most labs that do DNA work, you might never notice a small amount of PCR contamination. But in the context of deep sequencing that we do, a small amount of DNA really can mean thousands or millions of sequences in our data sets. So with that in mind, we have one room that is used for working with samples prior to PCR. And any machines that actually amplify DNA are down the hall, and the sequencers themselves are in the very end room. So one of our post-PCR rooms is the Mobix lab. This is where we do traditional Sanger sequencing and fragment analysis. So for this, we're currently using a 48 capillary ABI 3730, and this generates 48 sequences at a time. So you might think of this as kind of old-fashioned in a genomics lab, but that's really not the case at all. Often our clients just need a single sequence or a handful to answer their questions. So we actually run this instrument at least twice a week at this point. Now, contextually, you can think of Sanger sequencing really as a first generation of sequencing. So whether it was done with massive gels and radioactive labels or with capillaries and fluorescence, the fundamentals are the same. It's based on making fragments of different sizes and separating those by electrophoresis. And each combination of a sample and a sequencing primer just gives you one sequence, and that can be up to about 1,000 base pairs long. I'm sure you're all familiar with how this works and what the output looks like. So what do we mean exactly when we refer to next generation or second generation sequencing? There are actually many different approaches, lots of different instruments with unique chemistries, but most of them use some sort of sequencing by synthesis approach. And you can really interpret that quite literally. So we're finding out the sequence as we build a complementary piece of DNA. So if you compare that to Sanger sequencing, where we build many fragments of DNA from our template, and then we separate them and look at them after the fact to figure out the sequence. So live action sequencing, you might say this is. Now, the most important characteristic, though, is that these are high throughput technologies, so often called massively parallel sequencing. Instead of one sample giving us one sequence, we may be simultaneously generating thousands, millions, or even billions of sequences from any given sample. Now, historically, NGS also produced very short sequences in about the 25 to 150 base pair range. There are certainly exceptions to that now, but most systems still deliver relatively short sequences, I'd say below 300 bases or so. Now, the instrument you can see on the left here, this was really the first commercially successful NGS instrument, and that was made by a company called 454 Life Sciences. And I'm always proud to say that McMaster was the first Canadian site to have a 454, and that was thanks in part to the Ancient DNA group and their woolly mammoth sequencing. But as big of a deal as that was, NGS technologies change incredibly quickly. So here's an old picture on the left. That's the 454 in our lab in action. But on the right is our broom closet, where it's been living for several years. It's a very, very expensive paperweight, but that's the reality of these technologies. As of today, we have 3 main NGS instruments in our lab. That includes the Illumina HiSeq and MySeq systems on the left, and we also have a PacBio sequel. In the coming months, we will be adding an Illumina NextSeq. Don't worry about remembering all those names. I know they're confusing. But really, all of the Illumina instruments, they fundamentally work the same way, and they generate short reads under 300 bases. And the choice between them usually just depends on how many sequences that you need for an experiment. In contrast, the PacBio generates extremely long sequences. So typically we're aiming for about 10,000 base pair reads with that one. So before we can actually make use of these machines, there's a great deal of upstream work that has to be done to get the sample ready for sequencing. And we call this library preparation. And there are many, many different techniques that one can use depending on the nature of the sample. But the end products ultimately need to look a little something like what you see here. There's some fragment of DNA in the middle of the library, and that's the part you want to sequence. And then there are adapter structures flanking that on either side. And those are known pieces of DNA, and they serve as binding sites for our sequencing primers. Typically, we'll also incorporate some sort of barcode or index, you can see here. So what that is just a short piece of DNA that's unique to the sample. So we can combine multiple samples together on our sequencing run, and then we can bin them later on based on those sequences. So the top diagram here is just a rough idea of a typical Illumina library, and on the bottom is a typical PacBio library, which certainly are not to scale. This one is far, far bigger, the PacBio. So the next step, once we have our libraries, I'm skipping over weeks of work and QC, everything to make it just right. But then the next step is to load our libraries onto the sequencing instrument. So with Illumina machines, that involves a glass slide called a flow cell, which I'm showing in the top left. So the surface of the flow cell is covered with short primer sequences, which are diagrammed at the bottom. And those are complementary to the adapters that we're using. So they actually will hybridize and capture our library fragments when we flow them over the flow cell. From there, the fluidic system delivers sequencing primers, polymerase, and fluorescently labeled nucleotides, and it progressively builds a strand of DNA complementary to our library fragment. Now, what's important is that the nucleotides are modified, so the polymerase can only incorporate one base at a time, and it proceeds with repeated cycles of this chemistry. Add one base, followed by taking an image of that base that's been added, and repeat. So on the right is what the raw data looks like. So it's certainly not sequences, it's just images. So each speck of light that you see actually comes from one unique library fragment. And specifically in this image, you're seeing each fragment that has incorporated a G base at this particular cycle that we're seeing. So all of that gets extrapolated back based on the XY coordinates of each dot into a sequence. And as you can see, there are millions of them happening at the same time. So that was a very fast overview of how Illumina sequencing works. But let's contrast this to our PacBio machine, because a lot of the fundamentals are actually similar. We have a polymerase incorporating labeled nucleotides. But in this case, each fragment of library is bound to a polymerase molecule, and that polymerase attaches to a tiny well on what we call a SMRT cell, which is a picture at the left there. So in this case, the nucleotides are not blocked. The polymerase can freely incorporate nucleotides one after another, and the wells are monitored in a movie format rather than with sequential pictures. So also point out the barbell shape of our library at the bottom. So the images on top don't show it properly, but you can envision that this library actually would open up into a circle when you denature the DNA. And so the sequencing can proceed around that loop multiple times, and that allows us to increase our data quality every time it goes around that loop. Now, if you want really, really long reads, maybe you don't get around that loop many times, but there's a trade-off there depending on what you're doing. So let's talk more about why we use these NGS technologies. And I found over the years that a lot of people seem to equate NGS with genome sequencing, but that's really just one specific application. So essentially, kind of any project where you need a lot of sequences from a sample, there might be a way to do it with NGS, and perhaps it should be done that way. So besides whole genomes, we do a lot of sequencing that is targeted to particular regions of a genome, such as 16S profiling of bacteria samples. If you're not familiar with 16S, this is a ribosomal RNA gene. Some sections of it have a huge amount of variation, which can be used kind of like a unique barcode to identify a particular bacteria. So if you had a pure culture of a certain bacteria, you could PCR amplify the 16S gene and submit that to us for Sanger sequencing. And that one sequence would be all you need to identify the bug you're working with. But if you don't have a pure culture, what if you have a mixed community, sort of, for example, all the DNA isolated from a water sample or maybe from a poop sample, which is always a favorite of Farncombe researchers. In that case, the 16S gene is amplified much in the same way. But using NGS, we can actually get hundreds of thousands of different 16S sequences representing the many bacteria from that one PCR product. We can also prepare a whole genome for sequencing, but then selectively pull out regions of interest using methods like sequence capture. So that's what we do in the case of an exome data set. So in that case, researchers are only interested in looking at exons and variants that may affect protein coding. We can also use NGS to study DNA-protein interactions, like finding out where a particular transcription factor binds. We could do some basic epigenetic analysis, looking at methylation patterns with bisulfite sequencing. One of our most common applications, though, and increasingly so, is RNA sequencing. So in that case, looking at differential expression patterns across samples. So for instance, if I'm studying how a plant responds to different growth conditions, I could use NGS to count transcripts and figure out which genes are upregulated or downregulated in each of those conditions. But beyond individual genomes and transcriptomes, we can also perform similar analyses on community-type samples. So we call those metagenomes or metatranscriptomes if we're looking at RNA from a community. So this is what pushed me into the world of NGS, actually. So I was studying permafrost ecology. And with metagenomics, we could identify plants and animals and all sorts of microbes that were present at particular points in time, just from tiny amounts of soil. It would also, I don't have it on the slide here, but we could talk about COVID research. So as you probably know, basic COVID testing is just a qPCR assay. But NGS is used for looking at how those viral genomes are mutating over time and around the world. We do assist Andrew MacArthur and the Cancogen team with a lot of the sequencing that happens in Ontario. Another group at McMaster performs RNA sequencing to look at gene expression changes in lung cells that have been exposed to the virus. And there's another group still that's using targeted sequence capture to look for remnants of coronavirus genomes, and they're doing that in bat specimens from museum collections. Now, with all these applications, over the years, we've accumulated a number of companion instruments that we need for our workflows. So on the left, we have a nanodrop and a fluorometer. Those are used for quantifying DNA and RNA. The other instruments in the top row are Agilent's bioanalyzer and tape station systems. Those are used for checking fragment sizes. If you're like me, maybe you really hate running gels, so these are a great way to automate that. And the other instruments in the bottom row, we have a Covaris Sonicator and a Sage Science Blue Pippin. So these are used for DNA fragmentation and size selection, respectively. So even if you're actually not planning any sequencing, just know that we have these and we do run them as independent services as well. Another major platform that we use frequently is NanoString. Although NanoString does have a sequencer in development, what we actually have is their nCounter system. So it's not a sequencer, but it's a counting instrument, much more like a microarray, I would say. So in this case, target sequences are bound to a complementary probe, and that probe has a unique combination of fluorophores, as you can see diagrammed here, called the reporter, and those would link the the probe back to a particular target, and however NanoString has designed the assay. So the instrument actually can see these enormous probes, and it detects individual ones. So it's actually counting discretely single molecules of RNA in the end. And we mainly use this for gene expression analysis. So in the last few minutes, I just wanted to include a few slides about single-cell sequencing to follow Anna's presentation. And I agree with her that this is really important, and I think we're going to see a lot more of this type of work. So I'm just highlighting one company here called 10x Genomics and their Chromium instrument. There is a Chromium at McMaster, and that's in the Stem Cell Cancer Research Institute. Dr. Berg has graciously allowed us to use it, so we're gonna dive right in. We've just started on a couple projects actually. Now, this is not actually a sequencer. This is part of the upstream library prep process. So ultimately, we would prepare our libraries for llumina sequencing. And also important to mention that 10x is really rapidly expanding their product options. So you can use the system for a number of applications, but on these slides, I just have diagrams of their basic mRNA workflow. So if you think of typical bulk RNA sequencing, which we do all the time, but in that case, you'd be giving us a sample of RNA that's extracted from thousands of cells mixed together. And you can imagine that the data that we get from such an experiment actually represents just the average level of expression in all those cells. So in some cases, you may need to know how each individual cell or groups of similar cells are behaving. You could think perhaps of a tumor sample where you have different populations, or maybe immune system cells. And in that case, maybe an average of all of them might not be terribly meaningful for what you're trying to study. So until recently, you could spend a lot of time and resources separating those cells, separately extracting RNA from each of them, and then giving that RNA to us to make individual libraries from those tiny amounts of RNA. But as you can probably guess, that's extremely expensive and really for not a lot of data points in the end. But what the Chromium instrument allows is for us to take a suspension of cells and isolate them into tiny water droplets within an oil emulsion. So if you follow along the diagram that they've got at the top left there, the cells are coming in from the bottom. And each of those droplets also contains a gel bead. Those are the colorful circles in the diagram. And these beads are covered in primers that contain part of the llumina adapter that we'll need downstream, as well as a barcode. And that barcode is unique to that particular bead. So ultimately unique to the particular cell that it ends up in the emulsion with. So what you get out of the 10x machine ultimately is just this oily suspension of beads and primers and cells and all the reagents that you need for reverse transcription. And 10x calls these droplets gems. So you'll probably see that term a lot. And then from here, we would break open the cells, which are still isolated inside that droplet. And there we have everything we need to do the first part of an RNA library prep. So we can make cDNA inside that droplet. So after that, we'll clean up all the oil and we'll take the cDNA through a fairly typical Illumina library prep procedure. And then once all that is complete, every library fragment ultimately contains both the cDNA fragment that we want to sequence, as well as that 10x barcode, which is shown in green. So ultimately, we sequence both of them, and then we can group them back together. So all the cDNA sequences that came from a particular cell, and we can pass that on to someone like Anna for her expertise. So even though ultimately I only needed to make one Illumina library from that tube of cDNA, it's really more like thousands of single cell libraries in that one tube. So that's really incredibly powerful. And with that, that's all the slides I have for today, but I'd be happy to take any questions you may have. Thank you so much again.